Good morning and a warm welcome to all the participants present here. I, Ms. Anupama Tomar, on behalf of the Department of Microbiology and Biotechnology of Patkar-Varde College, welcome you to the webinar on recent trends in neurological research. It gives me immense pleasure to introduce our distinct guest speakers for the day, Dr. Jitendra Kumar, Officer, Glow Neuro, and Dr. Arup Acharji, Assistant Professor, Allahabad University. Before we begin, I would like to take a moment to introduce you all to our esteemed institution, Patkar-Varde College, founded by Chikitsak Samuh, named after Sir Sitaram Patkar, a distinguished judge of the Bombay High Court, and his wife, Lady Shantai Patkar. Sir Sitaram was deeply committed to advancing women's education and served as the Vice Chancellor of SNDT University for many years. Our College of Arts and Science began its journey in the academic year 1964-65 with the Commerce section following in 1980-81. To cater to the growing demand for career-based education, we introduced degree-level self-financed courses in 2000-2001. Since then, we have continually upgraded our courses and infrastructure to provide our students with the best learning opportunities. Many of our alumni hold prestigious positions across various fields. Patkar-Varde College holds a place of pride among educational institutions in Mumbai's western suburbs. We were re-accredited with a score of 3.53 by NAC in 2018, third cycle, and academic autonomy was conferred upon the college in the academic year 2019-20. As per the Education World India College ranking for 2020-21, our college was ranked 45th in India and 10th in Maharashtra. A brief introduction about our department. Department of Microbiology and Biotechnology was established in the year 2006. The department has been organizing Biofacet every year, started in the academic year 2012-13. Biofacet is a stride taken by the department to provide a platform to scholars, teachers, and experts from various fields of science and technology for academic interaction, elevation, and enrichment. It has also taken initiatives in helping students in extending their minds to diverse fields available in biological sciences. This would have not been possible without your support and participation. Thank you very much, dear participants. This year, we have organized Biofacet on recent trends in neurological research. As we all know that neurological research is crucial for understanding the brain, nervous system, and its impact on human health. It leads to better diagnosis, treatment, and management of neurological disorders, improving quality of life for patients. Additionally, research advances neurotechnology, unlocks human potential, and addresses global health challenges. Ultimately, neurological research transforms lives, improves healthcare outcomes, and revolutionizes our understanding of human brain and nervous system. And to elaborate more on this, we have the first speaker for the day, Dr. Jitendra Kumar Sinha. Presently, Sir is Chief Scientific Officer of Glow Neuro India, an organization focusing on brain awareness among general population, neuroscience consultancy to researchers and corporates involved in brain-related products, research and development. Sir has over 17 years of preclinical R&D experience in different areas of neurosciences, particularly involved in neurobiology of obesity, spinal cord injury, epilepsy, and neuro-oncology. He has ample research experience at various national and international research institutions of repute, including University of New South Wales, Australia, University of Verona, Italy, National Institute of Mental Health and Neurosciences, Bangalore, and CSIR, Center for Cellular and Molecular Biology, Hyderabad. He is a member of editorial boards of Frontiers in Aging Neuroscience and Matters. He is a life member of Molecular and Cellular Cognition Society, Indian Academy of Neurosciences, Nutrition Society of India, Society for Neurochemistry India, Society for Biological Chemists India, and many more. Sir has more than 50 international publications in reputed journals like The Lancet Neuroscience, etc. He has a cumulative impact factor more than 750. He has also published in 35 international book chapters. Presently, he is also editing 2 international book titles. There is a long list of awards and honors received by Dr. Jitendra Sinha, but in short, he has received 9 awards and fellowships from International Brain Research Organization, that is sponsor of our IBRO school also. Our prestigious awards are from Japan Neuroscience Society, UNESCO Society for Neurosciences, International Society for Neurosciences, Government of India, Korean Society of Lipid, and Arthrosclerosis, and many others. It is indeed our pleasure to have such an eminent speaker with us today. Before we begin with the webinar, I request all the participants to kindly be patient if any technical problems arise during the webinar. We will try to resolve all the technical glitches. Kindly post your questions in the chat box. It will be answered at the end of the session. We are sure that this webinar would certainly complement to your learning objectives. Now, I request Dr. Jitendra sir to address the webinar. Thank you.
Thank you, ma'am, for the elaborate introduction. And I also want to thank all the organizers, Dr. Pratibha Gaikwad, Principal, ma'am, Mrs. Ruchita Dalvi, ma'am, and all the patrons as well as the organizing committee for inviting me to this August gathering. And let's start the topic. Just a minute, I will upload here. I hope it is visible.
Yes, sir. Yes, sir.
So, a very good morning to all the audience and respected invitees and other members. So, as ma'am has already told about the neurological disorders, as why studying neurological disorders is very important, and how different research techniques or different research methods have resulted into uh, new kind of knowledge which has helped in treatment. And in many cases where treatment is not possible, the management of the symptoms at least can be done using the basics that we have learned over the years, over past many decades of research. So today I will be talking about some molecular mechanisms and the basics of different neurological disorders. So if we talk about neurological disorders, we know that it encompasses different or wide range of conditions that affects our brain, spinal cord, and peripheral nerves. These are the parts that make our whole nervous system. And these disorders can result from various structural, biochemical, or electrical abnormalities in the nervous system and lead to a variety of symptoms like paralysis, muscle weakness, poor coordination of different functions, loss of sensation, like seizures, confusion, pain, and also in many neuropsychiatric disorders, you will find altered levels of consciousness. So there is a long list of neurological disorders. More than 600 neurological disorders are known to us, but there are few common disorders that we often seen or we hear about them in day-to-day life. So I will be talking about the most common disorders, how they are caused, or what is the mechanism of these disorders. So if we talk about Alzheimer's disease, many of you would have for sure heard about it. And you know that this is a progressive neurodegenerative disorder that is the most common cause of dementia in older adults. So it is characterized by memory loss, cognitive decline, and various behavioral changes in the individual who is suffering from Alzheimer's. So it is caused by accumulation of beta amyloid plaques and tau tangles in the brain. I will be telling about these plaques and tangles in bit more details in coming slides. But in Alzheimer's, what we see that it when these kind of accumulation of different proteins or faulty proteins or plaques, tangles happen in brain, especially near or inside the neurons, it causes their functional disability, and due to which there is a loss of neurons and the connections or the synapses in the brain. And this happens particularly in cerebral cortex and certain subcortical regions of brain that results into Alzheimer's disease. The other one is Parkinson's disease, which is a chronic progressive movement disorder, which is characterized by degeneration of dopamine containing neurons in the substantia nigra. That's a region in the midbrain. And it causes like depletion of dopamine, which is a neurotransmitter, a type of neurotransmitter dopamine in the brain. And this causes the when there is loss and there is like different problems that are happening, the faulty proteins, we will talk about different genes also which result into faulty proteins and aggregates. These aggregates of or you can say abnormal aggregates of proteins make the Lewy bodies in the neurons that causes the degeneration of neurons, especially the dopamine producing neurons in substantia nigra. Just a minute, I will use a pointer. Just in case. Now we will talk about multiple sclerosis. So multiple sclerosis is a chronic autoimmune disease in which the immune system of the same individual, like the patient, attacks the myelin sheath, that is the covering of the neuron. Okay, uh, uh, so this leads to communication problems and between the brain and the rest of the body. And this, like when there is a problem with the myelin sheath, it causes demyelination. That means the myelin sheath is getting removed or getting destroyed, and due to which this communication, different communication problems, that is the signal transfer, becomes faulty, and in due course of time there is an axonal loss in the central nervous system, and this causes inflammation and formation of lesions or plaques in the brain and spinal cord as well. Then you would have also heard about epilepsy, right? So in epilepsy, what we know is it is again a neurological disorder which is characterized by recurrent unprovoked seizures due to abnormal electrical activity in the brain. So there is an imbalance of excitatory and inhibitory neurotransmission which results into this recurrent unprovoked seizures. And in due course of time, this also leads to different structural abnormalities in the brain. We have also heard about migraine, right? So migraine is a very common and debilitating neurological disorder which are characterized by intense throbbing headache, often accompanied by nausea, vomiting, and sensitivity to light or sound. And this like no different researchers have found that this happens due to activation of trigeminal system, and it releases it is caused by, you can say, due to the release of different inflammatory neuropeptides, and that even in long duration it can result into depression in the individual because of the throbbing pain and irregular uh, type of happening again and again in a very important period of time when we have some important work in hand, and such kind of pain happens that affects the whole uh, lifestyle of an individual. Although it is not considered as neurodegenerative disorder, but still a migraine, which is commonly taken as a headache or a very big headache, actually affects even different brain regions in long term, and it can result into neuropsychiatric disorders like depression. So we have also heard about stroke. So stroke occurs when blood supply to any part of the brain is interrupted or reduced to a very significant levels, and this results into the brain tissue from getting enough of oxygen as well as nutrients. So what will happen? The nearby neurons will start dying, and this cell death or loss of function happens in those affected areas where the supply of nutrients and oxygen is affected or prevented due to any type of interruption in the blood flow. So like you will hear about ischemic stroke, which is caused by blockage of an artery. So if artery is blocked, it will stop the supply of oxygen to that, and it will cause ischemia. So we call it as ischemic stroke. Whereas other type is hemorrhagic stroke, where due to bleeding or due to rupture of the blood vessel, some blood comes in the brain region, and that affects the regular physiology of those neurons in that brain region, and it affects, and in long term it kills the neurons there, and that's why it is called as hemorrhagic stroke. Also we although many few of us would have heard about amyotrophic lateral sclerosis, but it is also very debilitating, or you can say in long term the resulting into death. This is a neurodegenerative disorder, and it is also progressive in nature, also known as ALS or Lou Gehrig disease. On Lou Gehrig was a very famous baseball player who died in at a very early age of 38 years. So he was also having this ALS. In this disorder, the nerve cells in the brain and spinal cord are affected, and due to which the motor control in the body is lost. So when these motor neurons are degenerated, it results into progressive muscle weakness and atrophy. So when the muscles are not in control of the person, especially the respiratory muscles that are controlling lungs or the diaphragm, the person is paralyzed in due course of time, and that's how the death occurs. So many times somebody can ask, like, what is the importance of molecular research when we are talking about a neurological disorder, which is on a bigger picture? It's like it is debilitating and it's causing a problem to an individual as well as the family members or the society. Why we need to go into very details, and why we need to bother about very minute details of it, of that disorder, like why and what molecules, the small molecules that are not even visible, why to bother about them? It is very important. And why? Because these molecules actually play a role in modifying all the molecular mechanisms that drive our biological processes in our body. So the whole physiology is actually controlled by these molecules inside our cells, inside our bodies. And that's why molecular research is very important. So we will be understanding in the coming slides that how identifying different disease mechanisms are important, because it will help us in identifying the targets as where we need to look in the body, or which molecular targets we need to actually see or look for, and if it is faulty or not. And when we find that it is faulty, we need to develop different diagnostic tools, right? If somebody is having some disorder, just by outside look, we cannot find the exact reason as why it has been caused, or why it is causing different symptoms. So same symptoms can be in different disorders, but if we talk about molecular biomarkers, they are like no specific for certain disorder. Once you find that, that is called the target. And then if we develop different diagnostic tools, then we are able to find that that person is having this disorder. It also helps, the molecular research also helps in advancing drug discovery and development, where we develop different types of drug molecules, pharmaceuticals to treat, and also different other methods are developed which help in management of the disorder. It also helps in revolutionizing the personal medicine, which are the tailored therapies and pharmacogenomics, which helps at individual level. So same disorder can be precipitating in different ways or at different levels in different individuals, due to the genetic as well as different environmental factors like food or other pollution, anything that is there around us may affect at individual level. And that's why this genetic concerns and everything needs to be considered for a personalized medicine tailoring. Okay, so in this presentation I will be talking about molecular mechanisms of disorders to better understand what we know so far, how it can be applied, and some future directions on that. So first of all, like just to like no, while I was telling all this, I also felt like some basics need to be discussed, as what are the structure and function of neurons, the neurotransmission process, and few key brain regions that are affected in these neurological disorders we will discuss. So first of all, let's see a structure of neuron. A neuron is the basic cell of nervous system. There are other cells also, glial cells and all, but the main cell that transfer information from one part of the body to other is the neuron. So neurons are the fundamental units of the nervous system, and these are responsible for receiving. Okay, it receives, then processes and transmit the information throughout the body. They are the specialized cells that communicate via different electrical and chemical signals. More of the chemical signals are required, or are involved. This is how, and these chemicals are, or the neurochemicals are neurotransmitters mostly, which transfer information from one cell to other. When I say one, it is called presynaptic neuron, and the other one is postsynaptic. Next postsynaptic will become presynaptic for the next neuron. Okay, so it is like a chain of neurons, and the information is transferred from presynaptic to postsynaptic. So by now you would have understood that this part where the information is transferred from the pre to post is synapse. Okay, so I will tell one by one about these cellular components. So this part is cell body or soma. It contains the nucleus and organelles. It is the main part which is responsible for maintaining the cell's metabolic activities. You can see these dendrites, finger-like structures or branch-like extensions that receive signals from other neurons and conduct the electrical message to the cell body. This part is axon hillock, where actually the action potential is generated. Then that information or the electrical signal is transferred through the axon. What is axon? It is a long slender projection that transmits electrical impulses away from the cell body to other neurons. It can be muscle or gland also, wherever it is, these axon terminals are situated. So it is often insulated, like you can see here in the picture, with myelin sheath, which increases the speed of signal transmission. And this is known as myelin sheath. Okay, we will come to it, but first let's see the axon terminal. So axon terminal are located in the end of the axon, and it releases neurotransmitters. So all of the neuron was transferring electrical signal, but in the end it becomes a chemical signal to be transferred to the next neuron or muscle or like glands, right? So this here the release of neurotransmitters happen. Then we talk about myelin sheath, and these myelin sheaths are made of fatty layer that covers the axon, and it is produced, this fatty product is produced by different glial cells. So in the CNS or central nervous system, we have oligodendrocytes, okay, that produce this myelin sheath or make this myelin sheath, whereas in peripheral nervous system we have Schwann cells that make the myelin sheath. Also, when you see this myelin sheath, in between you can see some part of the axon is visible. So this part is known as nodes of Ranvier. Okay, so these gaps in the myelin sheath where ion channels are concentrated, and this helps in saltatory conduction, or you can say the jumping of, like, the electrical signal happens. And because it jumps from node to node, the signal speed increases to a very great extent, as compared to like channel. If it channel by channel, if it goes, it will be slower. So non-myelinated neurons are slow in conduction, whereas those with myelin sheath have been observed, or it is known that they due to this saltatory conduction, the speed of these neurons, or the transmission speed, is very fast in these neurons. If you see, so in previous slide we learned about the structure of the neurons. Here we will see the function of neurons, where you can see that these dendrites receive incoming signals from other neurons or sensory receptors. Then after reception, this information needs to be integrated. So signal integration happens of all these incoming signals, and it generates an action potential at the axon hillock. Okay, and this only happens when enough of signal is there. It is like all or none, right? It is not like whatever is small or big is there, it will happen. It has to cross certain limit of depolarization, then only this action potential is generated. Once it is generated, the signal is transmitted along the axon to axon terminal, and then signal output happens, where the neurotransmitters are released from these axon terminals, and that crosses the synaptic cleft to transmit the signal to the next neuron, also known as postsynaptic neuron. Now we will see how this neurotransmission happens as from the presynaptic neuron to the postsynaptic neuron. So we know that, okay, in this, let's suppose this blue neuron is a presynaptic neuron, this is postsynaptic neuron. So action potential, like it gets the information through our dendrites. Here, as you can see, the action potential is generated and then it is transferred. So action potential is initiated when the neuron receives sufficient excitatory signals causing depolarization of neuronal membrane. Okay, so it is called depolarization because normally it is in polarized condition. That means more of negativity is there inside the neuron, and when the signal comes in terms of positive ions, the negativity in the inner membrane of neuron decreases because of the positive charges, and this causes depolarization. So normally it is minus 70 millivolts, and then when positive ions keep coming, the negativity decreases, and when it reaches about minus 55 millivolt, the action potential is generated, and then it peaks to around 40 or 50, around 30 40 millivolts, and then it shoots back. That time it is repolarization happens. Okay, so that is the way the action potential is generated and how the depolarization happens. Once this is like this action potential is generated, the next step is propagation of action potential, where it travels down the axon, facilitated by openings and closing of different voltage gated ion channels. This voltage is again changed due to influx of ions and also outflux of negative ions. Okay, once it reaches to the end, then actually happens the neurotransmission, where it triggers when the signal is reaching to the end of the neuron or neuronal ending, axon terminals, right? All of these things are same. When it reaches the axon terminals, it triggers the opening of voltage gated calcium channels, okay, VGCCs. So when these voltage gated calcium channels are opened, there is a heavy influx of calcium ions, and which causes the synaptic vesicles. Here you can see, when the signal comes, these synaptic vesicles carrying the neurotransmitters, they are carried towards the end or the presynaptic membrane, where it fuses with it and releases the neurotransmitters that are contained in that vesicle. Okay, so once this is released, then what will happen, the neurotransmitters come outside in the synaptic cleft. This is the whole synapse, and this portion is known as synaptic cleft. So once these neurotransmitters come in the synaptic cleft, it also reaches to the postsynaptic neuron dendrite. Okay, on the dendrites you will find there are different types of receptors. So once this neurotransmitters reach these receptors, they can bind only to specific receptors. It is not like it will bind to every receptor that is there. It's like very specific. If you have learnt about enzymes and all, you will know that there is a specificity in there, and here also for neurotransmission there is a specificity of the neurochemical and its receptor. So they will bind to specific receptors on the postsynaptic neuron, and this binding can either excite or inhibit the postsynaptic neuron, depending on the type of neurotransmitter as well as the type of receptor. The same neurotransmitter can act as excitatory or inhibitory according to the receptor biology or the type of receptors. Okay, so once this happens, the last step in neurotransmission is signal termination. What happens in signal termination is that these neurotransmitters are removed from the synaptic cleft through reuptake into the presynaptic neuron, or different enzymatic degradation can be there, or it just or just diffusion away from the synapse can also be happening, due to which the neurochemical or these neurotransmitters are removed from there, so it is again refreshed for the next signal to come. Okay, so this is how neurotransmission happens. Now I will talk about certain key brain regions that are involved in these disorders. If you see Alzheimer's disease, uh, we know about Alzheimer's disease, how the memory and cognitive dysfunction is seen, but the key brain regions involved in Alzheimer's disease are hippocampus and cerebral cortex. Hippocampus, you can see here, is very critical for memory formation and often one of the first regions that are affected in Alzheimer's disease. It has also been seen that it affects the neurons of cerebral cortex, which, and the cerebral cortex, the kind of covering of the brain, it's not outside covering but the brain itself, this part is cerebral cortex. So it is responsible for higher cognitive functions, including the way we reason, our language, our perception about the whole environment, other people. All these things are controlled by cerebral cortex. And when the hippocampus, the where memory formation is happening and where the higher cognitive functions are being controlled, when these regions are affected due to the neuronal loss in Alzheimer's disease, you can easily understand that what will happen to the Alzheimer's patient. These functions, these very important functions are affected, and whole personality and important functions are lost in the patients. If we talk about Parkinson's disease, okay, so very important region is here, you can see substantia nigra. This is midbrain region, and here a certain group of neurons make a region known as substantia nigra. They are quite dark in color as compared to their nearby region, and that's why this nigra word. So it's like dark substance. So substantia nigra is a part of basal ganglia, and basal ganglia is involved in movement control. So any neuronal loss, and these neurons of substantia nigra produce dopamine. So these specific neurons are affected, they die, and due to the neuronal loss, there is it leads to dopamine deficiency. And when dopamine is not there, the function of basal ganglia is affected. And basal ganglia is known for regulating voluntary movements and procedural learning. So that's why you see that rigidity and different other movement problems in the Parkinson's patients. In migraine, as I told, they how deliberating it is there in our daily lives. So the key brain regions that are affected in migraine includes the trigeminal nerve pathway. Okay, so this pathway is involved in the transmission of pain signals, which sends the pain signals to the brain, and that these regions are affected, and it sends too much of signal, which makes headache and other symptoms of migraine. And due to the sensitivity of light or sound can be also there. The other key brain regions involved in migraine includes brainstem and hypothalamus. So brainstem plays very important role in regulation of our breathing, consciousness, blood pressure, heart rate, and sleep also. Whereas hypothalamus is very well known for its function in maintaining the homeostasis of the whole body. So these are the brain regions which have been found by researchers that they are affected in migraine, and accordingly drugs are developed to the molecular targets are found which are more, which are sending more signals. That much is not required, and that needs to be blocked. Okay, like in many painkillers or medications for pain, it stops the signaling itself. Okay, so this is for migraine. The other thing is amyotrophic lateral sclerosis, or ALS. The key brain regions involved in ALS are the motor cortex and spinal cord. And as I told, ALS affects the motor neurons that are responsible for voluntary muscle movement. And later on it results into paralysis of the muscles. Now we will talk about genetics, that are uh, genetics in neuroscience. We also call it neurogenetics. Here the importance of neurogenetics is that how different genes in neurons, because if they are affected, either they are producing more protein, genes produce protein and proteins have their functions, right? So if genes are affected, the protein can be produced in more amount than required, and that can be faulty proteins also, and can make tangles and inhibit or affect the whole physiology of the neuron, killing like in a long course it will kill the neuron because the other functions are not done. Or it can be the genes can be producing very less protein or no protein which are required for the survival of neuron. So they, like, genes are very important. And there are certain inheritance pattern and genetic mutations that have been seen in different disorders. Once they are, we will see that one why they are important to study, and once they are known, how we can work on them to get the benefit of drug discovery in that case also. So, when we talk about neurogenetics, it combines the principles of neuroscience and genetics to understand how genetic variations can influence the structure and function of our nervous system, as well as their role in different neurological and psychiatric disorders. So there are there can be genetic basis of neural development, where neurogenetics seeks to understand how these genes regulate the intricate processes of neural development, including the cell differentiation, migration, and connectivity. The insights into these processes help to elucidate how the nervous system is organized and maintained. And the second thing is genetic influences on behavior and cognition, where neurogenetics try to explore how these genetic variations can affect our behavior, cognition, and different personality traits. Also, we learn about hereditary neurological disorders, or the heritability of these neurological disorders, where many neurological and psychiatric conditions such as Alzheimer's, Parkinson's, epilepsy, schizophrenia also have a genetic component. Okay, so we will see a few of them in coming slides. So when we talk about genetic basis of neurological disorders, there are different types of these, like monogenic, polygenic, and multifactorial disorders. So in monogenic, as the name suggests, there is a single gene that is causing the disorder. So for example, Huntington disease. What happens that if a mutation or mutations in a single gene can result into a disorders, like in Huntington disease, which is caused by a mutation in HTT or Huntington gene. Okay, and that results into some forms of hereditary spastic uh, paraplegia, which are linked to mutation in spast gene. Okay, then we have polygenic disorders, and polygenic means many genes are, or multiple genes are involved in this. Each gene will contribute a small effect into the whole disease risk. So if there are 10 genes that are involved, all these 10 genes will add up to the bag of problems, and the whole bag of problems will be resulting into these polygenic disorders, for example schizophrenia and epilepsy, because there are multiple genetic loci which influence the development of these disorders. Then we have multifactorial disorders, where these disorders result from interaction between the genetic predispositions as well as the different environmental factors also. So conditions like Alzheimer's disease AD or Parkinson's disease PD, we call, are multifactorial. where both genetic and environmental components influence the disease onset as well as the progression of these disorders. So we have monogenic, polygenic, and multifactorial. So if we talk about monogenic disorders, I was telling you about Huntington disease. So these are caused by mutations in a single gene. We have to remember this much for sure. Okay, so these conditions are Huntington disease or HD is caused by a mutation in HTT gene, that is Huntington gene. It produces Huntington protein, and this results into it is specifically characterized by an abnormal expansion of CAG repeats. So a certain number, till certain number, it is normal, but when these repeats cross that number, okay, 35 repeats more than that, it will cause the full-fledged Huntington disease. And also if it is these repeats are too many, okay, like it's quite quantitative. If it is a lot, then early onset will be happening in the individual. If we talk about the pathophysiology of Huntington disease, this mutation in HTT gene leads to altered form of Huntington protein that accumulates and causes the neurodegeneration in basal ganglia. The inheritance pattern is autosomal dominant. If we talk about fragile X syndrome, it is X-linked dominant problem, which is caused by CGG repeat expansion in the FMR1 gene, okay, on the X chromosome, and it leads to reduced expression of FMRP protein, which is important for synaptic function and plasticity. And when the problem is there, it results into intellectual disability and different behavioral disorders. When we talk about polygenic disorders, we have schizophrenia as an example. So we have researchers have used GWAS or genome-wide association studies, which help them to identify multiple genetic loci associated with schizophrenia. So these studies have highlighted the polygenic nature of the disorder, with many small effect variants contributing to the disease risk. So when we talk about the pathophysiology, we have different hypotheses and models. For example, dopamine hypothesis, which is one of the most enduring hypotheses in case of schizophrenia, which suggests that dysregulation of dopamine neurotransmission contributes to the generation of this problem. And it is supported, this dopamine hypothesis is supported by the fact that different antipsychotic drugs, which primarily target the dopamine D2 receptors, act nicely for treatment of schizophrenia. Then we also have glutamate hypothesis. Emerging evidence suggests that if there is dysfunction of glutamatergic neurotransmission, particularly when it is through the NMDA receptors, it plays very important role in schizophrenia. So genetic studies have implicated genes involved in glutamatergic signaling to be one of the reasons. Then we have neurodevelopmental model, where schizophrenia is thought to result from abnormal brain development, possibly due to genetic and environmental insults during the critical periods of development of a child. And this model is supported by structural brain abnormalities that have been observed in the patients of schizophrenia. Okay, and there is also involvement of immune system, where MH series and other immune related genes have been observed to be involved in the pathophysiology of schizophrenia. The inheritance pattern is polygenic inheritance, and like no, it means that there are different genes that come, the product of those genes come together to result into the full-fledged disorder. Okay, now we have the third type is multifactorial disorders, where it's not only the genetic but also other factors. The environmental factors are also involved to cause that disease. So first we will talk about Parkinson's. In Parkinson's, please remember SNCA or alpha synuclein gene. Okay, so this alpha synuclein gene mutations and multiplications of this SNCA gene which encodes for protein alpha synuclein are associated with both familial as well as sporadic PD. And alpha nuclein uh, aggregation is the hallmark of Parkinson's disease pathology. There are other genes also like LRRK2 gene, then Parkin, PINK1, PINK2, DJ1 genes. All these genes, mutations in these results into Parkinson's disease. Specifically, when we talk about Parkin, PINK1, DJ1 genes, if these genes are mutated, it causes early onset of PD, which is like in at earlier age the patient will be having the symptoms of Parkinson's. The pathophysiology of PD involves progressive degeneration uh, of these dopaminergic neurons in the substantia nigra, and that affects the motor control in the individual, and that's why you see rigidity, tremor, bradykinesia, and postural instability in the patients. Also, it has been seen that the mutation in genes like Parkin and PINK1 affect the mitochondrial dysfunction. Okay, and this leads to energy deficiency or deficits and increase in the neuronal oxidative stress levels in the patients. Okay, further it causes neuroinflammation, and this chronic inflammation in the brain contributes to the progression of neuronal damage in the PD. So you can see the genetic problem or the mutation resulting into some problems leading to mitochondrial dysfunction, neuroinflammation, and already the genetic problem is there. So it further affects to increase the problem. So by this you can understand that how different molecular players are affecting the individual in these multifactorial disorders. Okay, if we talk about Alzheimer's, a very important gene, that is ApoE gene, that is apolipoprotein E gene. ApoE gene, particularly the epsilon 4 allele, is the most significant genetic risk factor for late onset AD. So there are normally two copies of epsilon 4 allele, which like no sorry, individuals with one or two copies of this epsilon 4 allele have increased risk of developing AD. Okay, so because time is less, I will try to bit, expedite the process. But yes, when we talk about the other genes like APP, PSEN1, PSEN or presenilin-1, PSEN2 genes, these, if mutations are happening to these, they are associated with early onset familial AD. Okay, APO is for late onset, APP, PSEN1, PSEN2 genes are for the early onset familial AD. Okay, so these genes are involved in production and processing of amyloid beta peptides. So, um, can I take some more time, or should I wind up?
Yes, sir. You can continue, sir.
Oh, thank you. So if we talk about the pathophysiology, like these accumulation of beta amyloid peptides, they form the extracellular plaques. Okay, so in the neuron, the outside region is, or outside the cell is extracellular region, right? So in that extracellular region, these amyloid beta plaques form the plaques, or amyloid beta peptides make the plaques because they are faulty and they make tangles. Okay, so they will be making a plaque, and this results into disruption of cell to cell or neuronal communication. Interneuronal communication is affected, and it also activates the immune responses using different glial cells and other immune cells there, which results into inflammation and neuronal damage. So we also need to know about the neurofibrillary tangles, that how it affects the hyperphosphorylation happens of the tau proteins in the inside the neuron, or due to the intracellular tangles made by these tau protein hyperphosphorylation. It disrupts the internal transport system within the neurons, and that causes the neuron to work in sub level, which results in due course of time the neuronal death is resulting out of it, and it causes neuronal death, as well as already the neuronal communication is faulty. So in due course of time the neuron dies, and nearby neurons also die, and it results into a significant level of memory loss and cognitive dysfunction. There in If we talk about the inheritance pattern, it can be sporadic or familial, but when we talk about late onset AD, it is mostly sporadic, but early onset familial AD is inherited as an autosomal dominant manner, often associated with the mutations of this APP, PSEN1, PSEN2 genes. Okay, so in the last section, common neurological disorders, where I will be talking about these four only, and how the key molecular players affect these different disorders. Okay, so the key molecular players in Alzheimer's disease are these. So we already know that AD is a complex on progressive neurodegenerative disorder, where a progressive memory loss and cognitive dysfunction or decline is happening. So when we talk about amyloid precursor proteins, or APP, it's a transmembrane protein that is cleaved by secretases to produce amyloid beta or A beta peptides, and the abnormal processing of this APP leads to accumulation of abnormal A beta, right? And this forms extracellular amyloid plaques, which is a hallmark of AD. Okay, in any paper that you read about Alzheimer's, you will find this. The hallmark of AD is amyloid beta plaques. Okay, then we also have presenilins, PSEN1, PSEN2, where these are the components of gamma secretase complex, and this is involved in cleavage of APP protein, okay, or APP, to produce the A beta. If there are mutations in these genes, then there is an early onset familial AD. Then we have apolipoprotein E, or ApoE. So ApoE, particularly epsilon 4 allele, is a major genetic risk factor for late onset AD. These days, many genetic tests and panels have come. If you go through those tests, if they find this epsilon 4 allele, they will tell that there is a possibility that you may get Alzheimer's disease in later part of life. So you can change your lifestyle. You take some kind of specific antioxidants or certain type of foods, and change your lifestyle in bit active mentally as well as physically, to help you not getting these disorders. Why I am telling this? That only studying in a lab is not enough. You should also understand that why we are studying all these molecular mechanisms. Once you know, you develop diagnostic tools, or some tools which can find the problem much before it actually precipitates in the individual. That's why these molecular research things are very important. Okay, so also there is a tau protein, which is a very important molecular player in Alzheimer's disease. So tau is a microtubule associated protein, or MAP, that stabilizes the neuronal microtubules. In AD, this tau becomes hyperphosphorylated. Okay, and this leads to formation of neurofibrillary tangles, or NFTs in short, that results into disruption of intracellular, inside the cell transport. The problem will be happening, and that causes the precipitation of Alzheimer's disease. There is a hypothesis known as amyloid cascade hypothesis that posits that the accumulation of A beta leads to series of events that culminate into finally neurodegeneration. That means the neuronal death. So APP is cleaved by beta secretase, also known as BACE1, okay, BACE1, and gamma secretase, resulting in formation of A beta peptides. Misfolded A beta aggregates like clump into plaques, and this results into the disruption of molecular function, neuronal function, and triggering neuroinflammation that you have known. Okay, so base 1 or beta secretase enzyme initiates APP cleavage, then gamma secretase complex, which includes PSEN1, PSEN2, involved in final cleavage of APP. Then this A beta aggregates form plaques that disrupt the synaptic function and induce neuroinflammation. When we talk about Parkinson's disease, we have SNCA, that is alpha synuclein gene, that encodes for alpha synuclein protein, which is a presynaptic neuronal protein involved in synaptic vesicle regulation, and finally the neurotransmitter release. Okay, so if there are any mutations or multiplications of this SNCase gene in the patient or the individual, it leads to misfolding and aggregation of alpha synuclein protein, and this forms Lewy bodies, okay, or Lewy bodies, which are pathological hallmarks of PD. Okay, remember the word Lewy bodies. So these are the hallmarks of PD. Then we have LRRK2 gene, which encodes for a protein kinase involved in various cellular processes, including the vesicle trafficking as well as cytoskeletal dynamics, which help in the overall functioning of the neuron. So if there are mutations in LRRK2 gene, such as G2019S mutation, it results into increased kinase activity, which is associated with neuronal toxicity and degeneration. Then we have Parkin, that is this uh, Parkin encodes for E3 ubiquitin ligase, okay, which is involved in protein degradation and mitochondrial quality control. So once any mutation is happening to the Parkin, it will lead to impaired mitochondrial function and increased oxidative stress. You have heard about lot about oxidative stress, that once it is there and if it is uncontrolled, it leads to the cell death. And that's how in PD, where the dopaminergic neurons are affected, this is happening, that the Parkin gene is mutated, which is affecting the mitochondrial function and hence increasing the oxidative stress, results into these dopaminergic neuronal death. Okay, then we have pink1 or P10 induced putative kinase 1. So if this is a kinase that regulates mitochondrial quality control and involved in mitophagy, the process of clearing the damaged mitochondria is known as mitophagy. So if there are mutations in pink1, it impairs the mitochondrial function and lead to increased vulnerability to again the oxidative stress. So again it will be contributing to the problem. Then this is actually we have DJ1, which acts as a oxidative sensor, oxidative stress sensor, and it protects from oxidative damage to the neurons. So if mutation will happen, you can easily understand that it will result into loss of protective function of the other components, and this again leads to oxidative damage and hence the neuronal death. Okay, so these are the molecular pathways. Like alpha synuclein aggregation happens and due to the misfolding. So this misfolded alpha synuclein aggregates form Lewy bodies, disrupting the homeostasis and leading to neuronal dysfunction and death. Okay, when we talk about dopaminergic neuron cell loss or neuron loss, it is a selective loss of dopaminergic neurons in the substantia nigra that leads to decreased dopamine levels in this striatum, and this result into motor deficits that are seen in the Parkinson's disease, right? So dopaminergic neuron loss is responsible for the hallmark motor symptoms of PD, such as tremors and bradykinesia. All these symptoms are seen in the PD. Also, we have mitochondrial dysfunction that we discussed, Parkin and PINK1 genes, right? So when they are impaired, they will be impairing the mitochondrial function, and oxidative stress will increase, and it will cause neuronal death. And when mitochondria is less, there will be energy deficits, and it makes the cell more vulnerable to different types of stressors, and that also causes cell death. So you can see all these different molecular pathways, they also finally, in one of the later stages, they contribute to neuroinflammation. And when it becomes chronic neuroinflammation, which starts mediating with the glial cells, the microglia and astrocytes, they get activated, and they these glial cells are trying to help the neuron to survive, but because they become so aggressive, they become activated in their function, their own secretions from microglia, activated microglia and astrocytes, they contribute further to the neuronal damage and progression of PD. So what will happen? The inflammatory processes will result into these neuronal loss, although they were meant to help, but they start killing the neurons, and this accelerates the disease progression in Parkinson's disease. So there are different pathways that are faulty, making the problem, and because the other cells, the activated microglia and uh, astrocytes, they were trying to help, but in overall resultant, the thing happens to be like it also start killing more neurons, and it gets accelerated. As I was telling you that when we talk about the genes that are implicated, HTT gene, which is Huntington. Okay, so the it is what happens that there is a CAG repeat expansion. One thing is that CAG repeat expansion happens. So normally it is less than 35 repeats in normal individual. Once it is more than that, 36, 39, 40, or it can reach to 120 repeats. Accordingly, the mutation will involve an expanded CAG trinucleotide repeat in this HTT gene. And in normal, as I told, it is less than 35, 10 to 35 copies. But when it is more, it will result into adult onset. And if it is more repeats, it will be juvenile onset. Like quite early in age, the person will be getting the Huntington disease. Also, there is toxic gain of function. What happens? That the expanded CAG repeats lead to production of mutated Huntington protein with an abnormally long polyglutamine or polyQ tract. This mutant protein is prone to misfolding and aggregation, and which is like no the which is toxic to the neurons, and this toxic gain of function also results into neuronal death. So there are different molecular pathways like protein aggregation, transcription, dysregulation. In protein aggregation, mutant Huntingtin protein, right, it aggregates into intracellular inclusions inside the cell. It happens, and these aggregates interfere with cellular processes inside the neuron. And once it is like interfering with normal processes, it starts to contribute into dysfunction, and finally cell death happens. So they can disrupt transcription, proteostasis, and also mitochondrial function in the neurons of Huntington's patient. So transcription dysregulation means when mutant Huntington affects the transcription by interacting with transcription factors, and hence it alters the gene expression. This dysregulation contributes to neuronal vulnerability and finally death of the neuron. Then we have mitochondrial dysfunction and oxidative stress. It is quite similar to what we have discussed previously. It contributes to neuronal damage and as well as apoptosis or apoptosis in neurons due to the mitochondrial dysfunction and increased oxidative stress. There is also excitotoxicity. What happens that mutant Huntingtin may lead to excitotoxicity by affecting the glutamate receptor function. Like excitotoxicity is like we have excitatory neurotransmitters, inhibitory neurotransmitters. When excitatory neurotransmitters are signaling through the receptor, very enhanced level of signaling, it results into excitotoxicity because it finally results into the death of the neuronal death. And this happens because there is excessive calcium influx in the cell, which the cell itself is not able to handle, right? Then we also talk about impaired proteostasis, the ability of neurons to degrade and clear the misfolded proteins that gets compromised in Huntington's disease. And this exacerbates the protein aggregation and toxicity, again leading to neuronal death. Okay, as the last disorder, ALS, these are the genetic insights where we learn that how these SOD1, that is dismutase and other, they contribute to the progression of disorder as well as the one of the causative agents. So this like if you talk about SOD1, this is first gene discovered to be associated with ALS, and it encodes for enzyme superoxide dismutase 1, which plays critical role in detoxifying the superoxide radicals in the um, in the neurons or cells, right? So this SOD1 mutation accounts for about 20% of the familial ALS cases, and these mutations lead to toxic gain of function effects. And then we have also when we talk about mutant SOD1 protein, it tends to misfold and form toxic aggregates which can impair the neuronal functions and contribute to the motor neuron death. Once the motor, because here when we are talking about ALS, you have to remember we are talking about motor neurons, because motor neurons are affected in ALS, and when these motor neurons are dying, the motor control is lost, and that's how the paralysis is resulting. Okay, so we have also other genes or factors like C9 ORF72, which is most common genetic cause of both familial and sporadic ALS. It accounts for about 30 to 40% of familial cases and around 10% of the sporadic cases. So it's also a mutation happening in this part, which results into toxic RNA foci and abnormal dipeptide um, proteins through non-ATG or RAN translation, right? So these toxic species abnormally disrupt the cellular functions, including various mechanisms like RNA metabolism, okay, and that results into cell death. Then we also have TARDBP or TARDBP, where this gene encodes for TDP43. Okay, it's a protein which is a DNA RNA binding protein, and it is very important. You would be knowing about TDP43. You would have heard when we study molecular biology in a general way, right? So this protein is involved in RNA processing, and mutation in this is found in a small percentage of like no both familial as well as sporadic ALS cases. And you will see that these mutant TDP43 protein mislocalize from nucleus to the cytoplasm, where they form the aggregates. And this mislocalization impairs the RNA processing itself, the splicing of it. And this, once all these things are like no disrupted, neurodegeneration is like no it results in that neuron. And when it happens in many neurons, it becomes when it increases, it becomes progressive. Then we have FUS, which is another RNA binding protein, and the gene is associated with ALS. And if there are mutations in FUS, it accounts for about 5% of familial ALS cases. What happens, similar to TDP43, the mutant FUS proteins mislocalize to the cytoplasm and form aggregates. And this disrupts the RNA metabolism and cellular homeostasis and causes cell death. Okay, so in the end, we will talk about this motor neurodegeneration, the molecular insights in the ALS, where we see that how this protein misfolding happens. So these misfolded proteins such as mutant SOD1, TDP43, and FUS that we just now discussed in last slide, they aggregate in motor neurons, leading to cellular stress and toxicity. Okay, so these aggregates interfere with their normal functions, cellular functions, including here specifically in ALS that we are studying. Mostly it is the protein degradation pathways, okay, that are affected. And due to that, what happens, that there is an accumulation of protein aggregates, and when it happens, it disrupts the proteostasis, the minute balance of these proteins, and this leads to neuronal dysfunction and death. When we talk about RNA metabolism and splicing, these mutations in the RNA binding proteins like TDP43 and FUS, it impairs the RNA processing and splicing transport, and that affects the whole neuronal health and results into neuronal death. When we talk about mitochondrial dysfunction, the SOD1 is the most important one to study, where the mutations in SOD1 and other related genes result into mitochondrial damage and dysfunction. And then again, apoptosis and cell death pathways get into picture. When we talk about neuroinflammation, we have to remember microglial activation and neuroinflammatory processes that contribute to the progression of ALS as such. So there are inflammatory cytokines and reactive oxygen species, also in short we call as ROS. Okay, so ROS, they exacerbate neuronal damage, and the chronic inflammation due to this accelerate the motor and disease progression. Okay, so with this all molecular insights into the most important disorders, I would like to wind up. And I would like to thank the organizers for inviting me to this very important platform. And I hope from all the audience that you would have learned something new, or if you already know, you would have revised what you already know, which will be helpful. Thanks a lot.
Thank you, sir, for such an interesting yet informative session. With this, we come to the end of session one of the webinar. I thank Jitendra sir for giving a detailed overview of the neurological disorders with common examples, along with the importance of molecular research in disease mechanism, drug discovery and development, and revolutionizing personalized medicine. Thank you, sir, for giving us the information on basic concepts of neurobiology as well as the different parts of brain affected in different neurological diseases. The concept of
Thanks.
neurogenetics was well explained through illustrative examples, that included the role of genes in different neurological disorders and the molecular mechanism of those genes in selected neurological diseases. Sir, we'll begin with the question answer session. Participants can post their doubts in the chat box or they can unmute themselves and ask their doubt. Sir, there are two, three questions which I like to take. The first question is, what challenges exist in the current molecular diagnostic approaches for Alzheimer's or any other neurological diseases?
Okay, so when we talk about Alzheimer's disease, as we discussed, there are certain genes that are affected and that are causing aggregates, intracellular aggregates, as well as extracellular plaques. The important thing is, like, although we are able to find it in different levels in the sections of these the brain sections of the post-mortem brain sections, it can be can be done, but the challenge is to find it at an early stage, and that challenge can only be solved when we are able to find the expression of these genes at a very early stage. So normally what happens when these genes or the affected proteins, when they are coming into picture, or when we are able to find it out, it is already late. So already neurodegeneration has started and progressed to a very high level, where at once we know that, although there are different management disease management paradigms where it can be kind of halted using different monoclonal antibodies type of medicines. Recently, just 1 week back, one paper came where a few new antibodies have come with new set of markers, biomarkers, where the physicians have to first segregate the patients and then do it. All this is happening after diagnosis, right? The main challenge in molecular paradigm is that how it can be found out. As I told, like epsilon 4 allele, one or two copies of it will be is considered as a probable futuristic progressive disorder to be happening in future. So if that can be done in a cheaper way, so the one thing is to find it out, and then other thing is these tests or these genetic tests or genetic screening tests need to be cheaper, and this can only be cheaper with more funding, where we have easy access to these tests, like we have seen in COVID-19 paradigm also, in the whole thing, that how it became, like it was discovered, different molecular diagnostic kits were developed, and it, like from 2000 rupees it came down to 50-60 rupees. And this only happens when the number of kits are sold in large number. Okay, developing phase, it will be very expensive, that needs more funding, and more research is required. So these are the main challenges that I see in AD research.
Thank you so much, sir. There is one more question. In neuroinflammation, which immune cells are involved?
So see, there are in inflammation in whole body, there are many immune cells. There is a long list of it. But when we talk about brain, the main molecular or main immune cells that are acting in a major form are microglia and astrocytes. So these microglial cells and astrocytes, they get activated, and when they get activated, their size increases. The genetic gene expression of certain genes might this microglial and astrocytic genes like GFAP, no, it's a very you can say traditional or very well-known protein, GFAP,
Hmm.
glial acidic protein. So the expression of it increases. That means more of it is there in these cells, which try to help the neurons by covering them, by providing them the support. But these are the what happens in neuroinflammation. This happens to be producing more than required, because it's like an exponential effect that happens, that one microglial cell or astrocytes, they start telling to nearby cells to also help, and that's how more of cells get activated, which further accelerates the neuroinflammation process, and finally they cause the neuronal death.
We have one more question. Can lifestyle changes or therapies help in managing neurological diseases?
Yeah, so it's a very nice question that we all need to be careful. So once a person gets into it, it becomes like we try to find the best doctor for getting the treatment. But the best thing that everyone should do is take care of their lifestyle. These days, most of the times, we all researchers, like these days students also have to learn so many things, so they stick to their computers or their iPads, mobiles, and all the time they keep sitting, due to which the whole physiology gets changed, right? We drink less of water, we eat unhealthy food, we do less of exercise. Even walking is very good exercise. When these kind of lifestyle changes that is happening in the whole generation, it results into accelerated way of neurodegeneration also. It's not only about the peripheral diseases, but also in diseases in terms of neurological as well as neuropsychiatric disorders, they get aggravated due to lifestyle. So it's very important to take care of our lifestyle and drink lots of clean water and take healthy food.
So one more question. How does protein-rich diet affect these neurodegenerative diseases?
It's a bit like no dicey question, where protein rich
Hmm.
diet, it depends like no which disorder we are talking about. In few of the neurological disorders, it is considered as high protein diet can be like no detrimental. So in there are different school of thoughts. So a few researchers say that vegetarian diet is going to help in improving the cognitive abilities, whereas other group of researchers have shown that certain proteins that are only available in animals, they are very much required for normal or healthy brain health. So still it is a unsolved question, that if this protein or high protein intake is helpful in maintaining the neuronal health or neurological health. So yeah.
Okay. Is there any research which is going on gene therapy approach for the treatment of these diseases?
Yeah, gene therapy is like in I just heard in few of the places gene therapy is in progress, but still it is in very early phase, where sometimes the results are in positive form, but also it has many aftermaths in the patients, where the patients are telling that it is resulting into some other problems which we don't have treatment for. So still it is in not in infancy. It has progressed, but we still lots needs to be done in gene therapy to actually get into treat as a proper treatment for the general population. So it's in uh, kind of advanced research phase.
So how can we assess the migraine in terms of molecular diagnostics?
So for migraine, there are certain molecular diagnostics, but these are very rarely available. So normally, physicians are using different physiological tests, as where the problem is happening, and symptomatic relief is the main uh, like uh, way of treatment that is going on these days. But yes, there are molecular or genetic um, connections that have been seen in migraine that I showed in the slide.
Sir, as there are many questions which are coming up in the chat box, so I will request you to share your email ID so that we can transfer this particular questions to you so that you can answer them as per your choice.
Yeah,
I,
sure.
I
I
request.
will. I will share my email ID.
Okay, thank you so much, sir. I
Thanks
request
a lot,
Anupam.
Kaushal.
Thank you, sir. I request Anupama ma'am to begin with the second session.
Thank you so much, Jitendra sir.
Thank you.
Okay, so now moving ahead, the next speaker for today's webinar is Dr. Arup Acharji. Sir is an assistant professor in the Department of Zoology at the University of Allahabad, where he is dedicated to advances in understanding the molecular mechanisms of brain diseases through proteomic approaches. His academic journey began at Banaras Hindu University, where he obtained his Bachelor of Science in Zoology, followed by a master's degree in zoology with specialization in biochemistry and molecular biology, and then completed his PhD in zoology. Throughout his career, he has been passionate about blending research with education, teaching undergraduate and postgraduate students, and guiding them through academic and research endeavors. His research spans several key areas, including infectious diseases such as COVID 19, where he identified pathogenesis factors and diagnostic biomarkers using proteomic techniques. Before his current role, he also worked as a project research scientist at a proteomics lab IIT Bombay and as a UGC Dr. D.S. Kothari postdoctoral fellow at BHU, delivering deep into the proteome profiles of excitotoxic rat brains and their modulation by herbal extracts. In addition to teaching and research, he is actively involved in collaborative projects, educational mentorship, and facilitation of international workshops, striving to inspire the next generation of scientists in the field of molecular biology and proteomics. Now, I would request to address the webinar. Sir, you can start.
Right. Good afternoon, and thank you, Anupama ma'am, for this kind in- introduction. Mumbai is my second home because much of what I would be talking today, I learned at IIT Bombay. And when Roshita ma'am sent out the invitation to me, the first thing it occurred to me, it would be nice to go back to the city that I call my second home. It's a very aspirational city, and I guess most of you who are staying there. Uh, I mean, the city has this air that you aspire to do more than you possibly can, and at the end of the day, since you're aspiring to do more, you end up doing lots more than what is maybe initially possible. So when I am talking today, I have just like the last lecture, I did not go into the details of what research I do. This is a small part of it, but mostly I would try to tell you what is proteomics, what is big data, and what is AI and ML, how you can use that, because the very thought of biology in us, it triggers in our brain this dicey thing that, okay, we have to memorize a lot of scientific names, we have to memorize a lot of pathways, um, lots of typical spellings which we really do not know how we would do it. So most of us kind of get fearful of this factor. But what modern biology is doing is it has taken out much of these information heavy stuff, and what we are doing today at the cutting edge of biology is primarily taking advantage of multiple technologies, which platforms which are there, and these technology platforms kind of digitize the data. Biological data has been digitized, and once a data has becomes digital, we can apply due to the development of so many advanced semiconductors and so many advanced uh, tools, primarily based on artificial intelligence and machine learning. We can do more than what we can actually can, just like the people from Mumbai. So I will just start off my presentation. Let me know if my screen is visible. Just let me know if the screen is visible.
Yes, sir, it is visible.
Okay, thank you. And you can interject me while I'm talking. Maybe the coordinator, the person who is emceeing this, they can take care of it. Put in your questions if you have. Raise your hands. I won't be able to see the hand, but if you think there is a doubt that needs to be solved then and there, please do interject me. So I'm used to it. That's how I conduct my classes, right? So, but do turn off your mics because it's the audio can sometimes be distracting. Okay, so how did we do science in the earlier days? Much of the genetics and that you would see. So there are departments of genetics and biochemistry, and all these classical genetics departments that came up in the 1960s and 70s and 80s. What they did was primarily they tried to catch a fish using a fishing rod. I don't know how many of you have an experience of catching a fish using a fishing rod, but the best, however hard you try, if you are using a fishing rod, you can only catch one fish at a time. And that's what people did earlier. They would mutate one gene, they will inhibit one protein, and then they will try to see what pathway it alters, what is the phenotypic expression, how does the phenotype change. And that's how much of the genetics and biochemical genetic and biochemical knowledge that we have gleaned from till the 1970s and 80s and even till 1990s. Now, how things have changed. With the advent of tools like genomics and proteomics, they cast a huge fishing net. And this fishing net will catch almost all proteins and it's their interactors and their genes. And we will try to analyze it as a whole. Just if I say that in the city of Mumbai, it's the financial capital of India. But do you think the entire finance is being done in Dalal Street? How many of you think that the entire finance of the country of India is being done at Dalal Street? I mean, you can let's keep it a dialogue, not a monologue from my side. So there is one view what people from genetics and biochemistry had was the entire finance since is done at BSc at Dalal Street, and that's what we will be doing. There is another view that even a villager who is paying GST in the farthest corner of India, maybe in Mizoram, in Tripura, when they're buying a matchstick, they are paying 5 paisa GST. Even that GST contributes to the development of the nation, that also contributes to the nation building. So this is what omics technologies try to say, that whatever be your gene or gene products, the proteins, they are all interconnected and all of them can have a impact. Now think of the COVID-19 pandemic. A man in China perhaps had a bad soup, and that kept you locked indoors for almost 2 years. So there's this huge ripple effect or the butterfly effect. So that's what we want to study in genomics and proteomics. We want to get all the connections. We want to understand all the connections, how they're connected, how they're interacting, and how that is impacting a disease process. So this is the central dogma of biology, and most of you would be knowing. So from DNA we get RNA, and from RNA we get proteins, and proteins become part of metabolic pathways, and we get metabolites. And um, DNA is primarily static, and what it is like is the Constitution of India, or the CRPCs, or the Bharatiya Nyaya Sanhitas. So it is the law. This should not happen, this should happen. But you all know that whatever in the law can be modulated, and whatever in the constitution is seldom followed. I mean, part of it is followed, and then part of it is open to interpretation. So that is what RNA is. RNA is a part of the genome that is actively transcribed. The entire genome is never transcribed. Now RNA tells us what appears to be happening inside a cell. It's again not the entire truth. RNA is like, perhaps the police stations and the police force. They are traveling around this. They are going around the roads, hoping to prevent crime. Proteins are what makes it happen. So it's that part of that police force who is actively chasing a thief. Not all police people are chasing thieves, right? I'm trying to give you analogies so that you can understand. So our constitution tells that to have law and order, you should have a police force. So RNA is that part of the constitution which deals with policing. Now, in that policing, not all police people are searching for criminals. Few are just sitting waiting for an FIR to be filed. But once an FIR is filed that, okay, I lost my watch, or I lost my mobile phone, a part of that police team starts working. Now, not if 10 people are working, not all would be successful. And after 20-25 days, you get back your mobile phone, and you see at the you receive a call from the police station that your mobile phone, we have retrieved it. And there are only two, or there is an inspector and a havaldar who is handing it over to you. So those are those metabolites, those people who actually did the work. And so this is the central dogma of biology, and you have to understand, and that makes proteins interesting. So again, giving a comparative example. So if you think of a traffic intersection and a parking, genome is very constant. It is not changing for a moment. The genome you are born with, apart from those mutations that Sir was talking some time back, even those mutations perhaps you are carrying. Uh, Proteome, on the other hand, is dynamic. The proteome you have today in the morning and right now in this afternoon would be definitely different. Again, by night time, the proteome of your particular cell would be different, and it keeps on changing. So think of this dynamic traffic intersections of Mumbai. The moment the traffic light changes, the cars are whizzing past. And again, if there is at night time, there are no cars, it's completely empty. Then it peaks during office hours. So proteome keeps on changing. And this change can reflect and can tell us stories about what is happening inside a cell. So what is proteomics? So Mark Wilkins in 1994, he was doing his doctoral studies at Macquarie University in Sydney, Australia. And he was going through the term genomics. The genomics as a term had just come up, and people were talking of the entire gene that a cell has, the entire genome. So genome and genomics, the term has already been coined. And Mark Wilkins was studies on the protein, and what he wondered, okay, if I am studying the entire proteins of these cell, can we not call it proteome? So this is Mark Wilkins. Obviously, this is not from his PhD days. He is now a very senior professor at Australia. So proteomics, he described the entire complement of proteins, including modifications produced by an organism or a system. Now, you have to think of this in a way that proteome can differ based on the baseline we are setting. If I'm talking of the proteome of a cell, if I'm talking of single cell proteomics, then it is a different set of proteome. If I'm talking of the proteome of an organ, like brain proteome, then it is different. And again, if I'm talking of human proteome, Again, I'm changing the relative scale, so stuff change. But as I said, why should we study proteomics? So first point is, proteins are the workhorses. They are the guys who are doing the work, right? Just having a set of laws in our constitution and telling that, okay, we should have a very good police force, won't solve the problems of crime, right? You need to have foot on the ground. So this foot on the ground are the policemen who are actively scanning the streets uh, for thieves and criminals. So these are these proteins, the molecules which do the work. So they're the catalysts and maintain the metabolic processes in the cell. Next, they are structural elements. Okay, say they are all the cells that we see. They have to be. They have a cytoskeleton, and all of you would appreciate the fact that all cytoskeleton are primarily proteins. So they are structural elements. Also, think of the signals, the cellular signaling that's happening all the time. The all signaling molecules are again proteins. They are receptors. So if you have a signaling molecule, you need to have a receptor, and all these receptors are again proteins. Intracellular signaling components, again proteins. If you want to determine a gene is being going to expressed or not, so think of those transcription factors. They are again proteins. Think of DNA replication, DNA recombination, RNA splicing, editing. All these processes, you would again notice, again there are sets of proteins who does these things. So point is, if you can study the proteome, you get a much better understanding of the stuff that is going on in your body. And if you see the current state of proteomics as a research topic, you would see there is a huge spike in the number of papers and number of and as time has progressed. So up till in the year 2000, you will see there are very few people, right? You can just, I don't know. Let me know if I can see your pointer. Can you see the pointer? Anyone? Can you see the pointer?
Yes, sir, we can.
Yes,
Can
sir.
you see my cursor? Am I audible?
Yes, sir, you are audible.
Hello.
Yes, sir, you are audible.
Yeah, you are audible. You are audible, sir.
Hello.
Arup, sir, you are audible.
I'm not sure if I'm audible. I mean,
Sir, you are audible.
you need to speak while I'm talking. You need to speak, else I won't be able to listen to you. I cannot see the emojis right now. So, whosoever is coordinating, can you just have this? Can you just act as a bridge? Who is coordinating this stock? Pushkar, you need to be. You need to use your mic when I ask him,
Sir,
because
sir, you are audible.
right, because was my cursor visible? Pushkar,
Yes, sir, it
please
was visible.
turn on your mic. When I'm on full screen mode, I can't see the emojis, nor can
Okay.
I see the chat.
Okay, sir.
So,
Okay, sir, got it.
please make sure that right. Oh, you gave me a scare. I thought I got disconnected. I'm speaking to a wall. Right, Pushkar, I will be going back to you. So
Okay, sir.
let me know if you can see my pointer,
Yes, sir.
and you can see the laser
The
pointer, right?
pointer is also visible.
Okay, I'm audible and pointer is visible. Fine.
Yes, sir. Yes,
Good.
sir.
Thanks, Pushkar. So you see the number of publications that have used the term proteomics, they have grown incrementally over the period of time. So point is, more and more studies are using proteomics as one of the ways to understand the biology of the disease. And this is a very nice study where they tried to see, many a times you would see people showing that, okay, my RNA expression went up. People are doing transcriptomic studies to see, okay, there is more RNA, and this is I mean, the more RNA you have, the better. So RNA, studying RNA for studying the biology of any event that is happening inside, apart from the regulatory RNA part of it, it's kind of misleading because RNA is always a proxy. Many of you would appreciate the fact that after transcription, translation, there are transcription, there are post-transcriptional modifications that happen. Like you have siRNA, you have, there are so many kinds of regulation that happens at the level of RNA, that even if an RNA is produced, it's not necessary that it will impact the phenotype of the cell. Next, even in proteins, we must appreciate that proteins sometimes work only when they are there are post-translational modifications. Like suppose there is a protein that needs a phosphorylation to get active. Now, even though the RNA might be produced in huge amount, if the protein is not phosphorylated, we do not know whether the protein was actually working or it was just standing there. It's something like having lots of policemen, but they're all off duty. A policeman can only enforce law when they are on duty. Now, a police person is not on duty all the time. They're on duty for 8 hours, maybe 12 hours under exceptional circumstances. So, if a crime is happening, a police person who is off duty or who has just retired from service, they can do nothing. They are just civilians. So proteins that so we need to study proteins, proteomics, just because they help us understand how cells work. And if you have done this study, and this what they did was they studied the you know correlation between RNA abundance and protein abundance. And if you see for a specific protein pathway, the correlation is comparatively higher than between protein. So between protein for a given pathway between Caenorhabditis elegans and Drosophila melanogaster, you would see that RNA for a given metabolic pathway there is only 0.47 correlation, and transcript again is 0.2. To 0.79. A perfect correlation will be 1, and zero correlation will be 0. So between 0 and 1, if you see, 0.8 is a respectable number. And this is what they did in this study. They did for the same pathway, they wanted to study what is the correlation between the protein and transcript. So we share different pathways with all other species, right? Although we are very different from a bacteria, but there are few basic pathways that a bacteria and a human has in common. So this study wanted to see whether the common pathway has common transcriptome or common proteome. And what they found was the proteome was more likely to be more common. And what kind of proteomics experiments can you study? So you can do discovery proteomics, and these are used for exploratory investigations. It's something like you take a mixture of cellular lysate, you take a group of cells and you want to study, okay, this group of cell, when they are sick, what are the proteins they are expressing? These group of cells, when they are healthy, what are the proteins they are expressing, right? So this is for exploratory investigations. You want to study if there is a difference in the expressed proteome, and from those differences you would try to understand or extrapolate the findings that, okay, since there were these differences, we can tell that, okay, these are the pathways that got disturbed. Another is targeted proteomics, where you try to see whether there is a protein specific A, B, or C is present in my sample, right? Especially true for biomarkers, or when you do a Western blot, or this kind of stuff, or even with mass spec, you can do RM, MRM. So you want to see a specific protein, if that specific protein has gone up or down. And this is only for, it's a follow-up of these discovery proteomics most discovery proteomics studies need to be validated using targeted proteomics. And as just we discussed, you would see the genome, although it's approximately to 22,000 genes, the more it becomes complex at the level of transcriptome, and it becomes very complex after we take the post-translational modifications into account. And that is why studying proteomics becomes very important, and it helps us to study variations. Again, one more example is, suppose a human gene, it can have two mRNAs. You have heard of splice variants, right? So there can be a splice variation, and if you see in the splice variants, there is a small variation here. Otherwise the proteins are almost similar, right? Then this RNA will have, again, site specific, so it can have a single nucleotide polymorphism, or it can have glycosylation or phosphorylation sites where the particular protein would be. So what you starts off as a single human gene, you get three different proteins. So this is just a random example. This paper came up in Science, the Human Proteoform Project. Proteoforms are just isoforms of the proteins that we express. And I must tell you, given the technologies that we have today, there is no possible way to study the entire proteoform. Although we know the entire human genome, entire proteoform is years away. We have a basic idea of the proteins that we express, but not all the proteins that we express. So this is one area of research which is completely empty, and we need young people like you who are studying in your graduation and post graduation, and you can pick these studies up for your PhD and do further research and try to tell us what are the different proteins or proteoforms that we have as a single human. Maybe you can start off with a single cell. Maybe you can tell, okay, a neuron throughout its lifetime will express these 25,000 types of proteins. Maybe there are 5,000 proteins, but each protein would be, some would have a glycosylation site, some would have a phosphorylation site. The same protein can have 10 phosphorylation site or 2 phosphorylation site, and the phosphorylation site again will determine whether how the protein will act. So proteome is larger than the genome, and of course we now know the reasons. So why, again, proteomics, what it helps us, it helps us decoding protein interaction and functions. So proteins not only act solo, they act as a team, sometimes like a receptor and ligand. Now these receptor ligand interactions can change the process of how a biological function or a disease process goes ahead. Um, Sometimes it can also help us understand genetic modifications. So suppose you have a GMO. There is a huge noise regarding genetically modified organisms, but if you can really study the proteome of a GMO and a non-GMO food, you can tell us in for specific by specific reasons that, okay, in the GMO food, these five proteins are expressed extra, and these five proteins have these five definite roles, and that can make acceptance of genetically modified organisms much easier. Disease mechanisms and personalized medicine, of course, is perhaps the most important part. Just as sir was telling, Jayan sir, that okay, we might have different uh, proclivity for a different disease. I might be more prone to Parkinson's or Alzheimer's. We need to find biomarkers of these diseases, and that can help us in personalized medicine. So there are studies that are going on around the world where people are trying to see, by understanding the proteome, if you can predict the arrival of a disease. And people have done good studies from the Matthias Ulen lab in Sweden. They have started given a very strong set of predictors for cardiovascular disease, and you would see there is a huge rise and spike in cardiovascular diseases that many people are having heart attacks and silent heart attacks, and all these cardiovascular diseases are big challenge, because we really do not know whether I will have a heart attack today or tomorrow. But proteomics, as Matthias Uhlen's lab have shown, you can actually predict by a set of biomarkers. The more that biomarker goes up, 5 years down the line, a person can have a cardiac arrest. So if I can warn a patient 5 hours, 5 years prior, that okay, these are the biomarkers that are shooting up and you are becoming more prone to cardiovascular disease, you can have some disease modifying interventions, you can have some medicines, you can take some lifestyle, you can do change your lifestyle, you can change your the food you eat, the amount of exercise you do, and stuff like that. And also, as I said, predictive medicine and personalized medicine, all these stuff are becoming real now. This basic chart shows us what are the various methods by which you can do proteomics. So one is expression proteomics. Now expression proteomics is primarily what we discussed right now. Okay, we study the all the proteins that are being expressed, and you do what is known as proteomic profiling. You compare the proteome in pathophysiological processes, you identifying the disease specific biomarkers, and you try to do therapeutic targets. Also, you can do structural proteomics, where you try to study the structure of a protein, how the structure of the protein changes with the different complexes. If I'm talking of a neurological disorder, we are talking of tau proteins, we're talking of um other Huntington's proteins. So these proteins, how they are interacting with other proteins in the brain. Can we do something so that the structural binding that is actually leading to neuronal death can be prevented? Then functional proteomics. So functional proteomics can be again thought of subcellular proteomics. So subcellular proteomics means how these proteins are moving around in the cell. Are these proteins nuclear or cytoplasmic or membrane bound? Are these proteins changing their subcellular location based on a disease pathology? Now you can take, for very nice example, you can take in the brain we have the synapse, and from the synapse we can extract synaptosomal proteins, means proteins that are precisely involved in the synapse. Now you take two disease, maybe Alzheimer's and a common human brain, and you see what are the proteins that are differentially expressed in the synapse of an Alzheimer's patient, or maybe in epilepsy. You can change, you try to see what in an epileptic rat model, what are the synaptic proteins, are they different from what they are in a normal animal, and that can give us understanding of how a synapse is working. Another is metaproteomics, not of much use for you guys right now, but that is we know that we have lots of microbes within our body, and they are good microbes in our gut, in our lungs. We are home to lots of bacteria and other microbes like fungi, and they produce metabolites, and they keep us healthy even. So by studying their proteome, we can there is now new studies that people are trying to indicate that the microbial profile, there's a huge study on the gut brain axis where people are talking of the microbial profile of your gut can influence your mood, can influence your susceptibility towards the disease. In fact, multiple neuronal disorders like Alzheimer's, Parkinson's, epilepsy, Huntington's, they all have shown strong correlation with the microbiome in the gut. The microbiome in the gut changes. So it's so right now it's a question of who came first, right? The chicken or the egg? So did the microbiome change and the disease happened, or the disease happened and then the microbiome changed? So people are still trying to understand this cause and effect, but proteomics studies like metaproteomics can help us understand the microbial profile. So also sometimes what we can do is we can do proteogenomics, and this is very important for cancer now, because you would appreciate the fact that cancer is a cell that has gone rogue. And this cell right now will be expressing lots of proteins which are usually not produced. And these proteins are foreign proteins to our immune system. Now, think of this. If I can identify correctly the foreign proteins or the non-native proteins that are being produced by a cancer cell, I can effectively train my immune system to act on all those cells which produces this aberrant protein. And this is what people are doing. In IIT Bombay, Professor Rahul Purwar, he was one of our collaborators while I was in IIT Bombay. They have developed something known as the CAR T cell therapy, and CAR T cell therapy is one of the most foolproof as well as most efficient way of treating cancer. It's just like having a highly trained sniper and killing off the criminal using a very long range bullet. So you train your immune system using proteogenomics. You try to find out which are the aberrant proteins, find the aberrant proteins which can elicit an immune response, and then you produce what is essentially a cancer vaccine. So if you see, the proteomics has huge ramifications. The if once you know the technique, once you know the technology, you can apply it to multiple diseases. And how do we do proteomics? So this is a basic workflow. I won't get into too much of details on this slide itself. And please do let me know how long I can speak. It's 107.
Yes, sir, you can continue.
Right. So what's the time, like 2 o'clock?
Yes, sir.
Fine. So if you see, we have samples, from samples we extract proteins, and then we can do gel based proteomics where you use gel platforms like polyacrylamide gels. You can do DIGE, that is differential in gel electrophoresis, or you can do a simple 2D, and then you can study the proteins that you found out using a MALDI-TOF. Or you can do gel based proteomics where you you digest the proteins, you do alkylation and tryptic digestion. I'm skipping the techniques part. And you can do LC separation, and based on LC separation and ion mass, you can detect the different proteins, and these proteins are finally found by something known as database searching. So using this, you can find what are the proteins that are being expressed in our cells. What are the major areas currently that we are studying in proteomics? One is mass spectrometry based proteomics. So this is the area most commonly associated with proteomics, and we use instrument known as mass spectrometer. And using mass specs, we can determine all the proteins that are being expressed. Then array based proteomics. Here we try to find proteins using a microarray based approach. Um, just in the speaker just prior to me, Dr. Jayant, he was talking about antibodies as biomarkers. Now, array based proteomics is excellent for this, because all you need to do is search for any antibody that is present in your blood and think of it. Why do we produce antibodies? Antibodies are produced when the protein is foreign to us. Why can a protein go foreign to us? Perhaps this protein is usually not expressed in the body. It can be a cancer cell. So these biomarkers are very easily identified using microarray based proteomics. And of course, the final part is informatics. And much of the data that is being generated in proteomics is technically big data. I will come to it, what is big data. But big data needs computers to analyze. We humans can never analyze that. So informatics based technologies are very essential to understand, what is needed in the cell, what is stored, and how we can study the proteome of the cell. Now, coming to mass spec, just to give you a very basic understanding of what is a mass spec, we take the sample, and this sample is primarily peptide. We take the proteins and then we digest the proteins using enzymes such as trypsin. Then we ionize these proteins, and ionization can be done using ESI or electrospray ionization or MALDI. Then you send it to a mass analyzers, and what these mass analyzers do, they separate the ions based on mass to charge ratio, and sometimes it is by time of flight, sometimes it is done in an orbit trap. Then the detector detects, okay, these are the ions that we found in the sample, and based on the signature of their mass to charge ratio, we determine, okay, these are the proteins that are being expressed. So MS based proteomics approach. So this is the entire pathway that happens. So you take protein, so this is a pure protein, you digest it, make peptides, you put it up in the liquid chromatography column, right? Then it goes to the electrospray ionization. You have molecular ions. You do the first MS, that is single M by Z. Then you dissociate these, you break down these ionic peptides into smaller fragments. You do the second MS. This creates the MS spectra. And this MS spectra is then understood not by a human but by a machine that we call the search engine. And based on the search engine, what they give us is they tell us whether this protein has any post translational site, what is the protein quantification, what is the protein ID. So this is the computing part, this is the mass spec part, this is the wet lab part, and this is what goes on in a typical proteomics lab. And what can contemporary proteomics do? In a single tissue or cell, it can routinely identify almost 3,000 proteins. Think of it, 3,000 proteins in a single small drop of protein lysate. It's a very small drop. It's like 1 microgram of protein. It's very, very less. From that, you can identify 3,000 proteins. And I'm talking of very cheap instruments. Now, if you go for a very costly instrument like the timsTOF Pro, it costs around 15 crores. And that instrument can tell you from the same droplet, it can give you data for 10,000 proteins. 10,000 proteins that are available in that cell, it can identify. And it is sensitive, and it's very sensitive. It does not only tell what protein is expressed, it can also tell you how much of that protein is present. It can also tell you the post-translational modification, whether the protein was phosphorylated or glycosylated. It can tell us about that. Also, it can help you understand whether the proteins are interacting with each other. So this is the amount of information that you get, and that is what brings us to the big data. Now, if you think of 5,000 proteins that are around 50 patients. So let's take an example. You are studying an Alzheimer's patient, and you have, or epilepsy patient, and you know epilepsy patients often undergo lobotomy, where a part of their brain that is causing the seizures are surgically cut off. Now you want to understand as a scientist that, okay, what went wrong with those part of the brain. Now you are taking over a period of this is a long project, maybe it takes 2 to 3 years. You have collected 100 patients brain sample, and they all have similar type of epilepsy. And you have also taken 100 patient brain from other patients, like maybe a patient who had undergone brain surgery for cancer removal. They do not have epilepsy, but you have taken brain from the same region, right? Maybe it's a frontal lobe or the amygdala or any specific region of the brain. So you have to do that control experiment. And or perhaps you have taken patients from, you know, autopsy samples. Sometimes many patients, many people, sadly, they pass away due to a road accident, or maybe they had some other trauma due to which they passed away. You can take those samples as well if your institute has those ethical approvals. Now you want to identify. So you have 100 controls, which are, they were healthy brains, and you have 100 patient brains. Now you want to understand what are the proteome profile. Now 100 multiplied by 100, what number do we come to? 10,000, right? And now you, in these 10,000 samples, you identify 5,000 proteins on an average from each of these samples. Now this data has become literally very, very huge. How do you make sense of this data? Because now it has become biological big data. And what is biological big data? It refers to the vast amounts of data that is generated in omics field. And it can be generated not only in proteomics, but also in genomics, transcriptomics, and this entire volume and variety of data presents its unique set of challenges and opportunities. So, and why should we study why big data matters in proteomics? Because proteins interact in complex networks, we just discussed. It's just not Dalal Street, the BSC Sensex that is controlling the economy. Economy is being controlled by a small farmer in maybe Assam, a shepherd in Kashmir. They are also contributing to the economy by their part. They are also consumers, they are also producers. They are producing milk, and maybe the shepherd is producing milk and uh, this pashmina wool, and they're consuming internet, they're consuming electricity, so they also becomes part of this greater economy that we want to study. So Dalal Street is just a small fraction of it. So we, as a omics scientist, we will try to understand this entire complex network. So big data will help us to map these networks, and by using large, analyzing these large proteomics data sets, we can identify biomarkers for the disease. We can understand whether a particular disease have gone drug resistance, or also we can identify new therapeutic targets, because now we know, and I will tell you about this new therapeutic target for the study part of the later part of my study. So it allows us to see the big picture, right? A more holistic and accurate, and it gives us a more accurate and holistic insight, and which is very important in today's science. But as I said, these are the opportunities, and there are lots of challenges. First is data complexities, we just discussed. For, if you have 200 samples, 200 patients, 100 control, 100 diseased, and you get 5,000 proteins per patients, the data has become very complex. Then storage and management. A average file from a mass spec that contains 5,000 proteins will be around 5 GB. Now for 5 GB file, you need to have, so 5,000 GB, or around 5 terabyte storage you need to store only for your patient data. If anyone has a question, you can ask.
No, sir, you can continue.
Yeah. So you need 5 terabytes for storing your patient data, 5 terabytes for storing your control data, and maybe while you analyze these data sets, you will produce more data. So we are not talking of mbs or kbs, we are talking in terabytes and petabytes. When comes, and it needs a lot of storage management softwares. You have to take a backup so that you don't lose your file, and this is really a pain in the neck. Once you start working on big data, this becomes really challenging, because one hard drive crashes, you lose data that took almost 3 to 4 years to collect. So you would need, people nowadays have shifted to cloud storage, where people subscribe to cloud space like Amazon Web Space and stuff like that. And analysis, that's is the toughest part. How do you make sense of these 50 lakh proteins that are expressing, because there is also something known as individual variability. So although I might have epilepsy also, and you might have epilepsy, you and I are not the same. Our genome is different, the way we were, our lifestyles were different. So extracting meaningful insights becomes very tough. These are the big challenges. And here, how do we solve these challenges? We use advanced computational tools and algorithms such as AI and machine learning. These are umbrella terms, but there are many terms like random forest classifiers and support vector machines. And all these tools help us in making sense of the data, because and also we can use AI to simulate these protein complex interactions. Many of you must have heard about AlphaFold, that has been touted as the best use of AI. So AlphaFold is a software that can predict the structure of a protein, and even if there is one small mutation in this entire protein, how the structure changes, it can faithfully, almost with 99% accuracy, tell you. And to put things into perspective, the very same data that you get in less than 5 to 10 minutes would have taken you 5 to 10 years in a normal lab, in a wet lab. So, in a wet lab, you have to purify a protein, get it into crystal structure, then you do X ray crystallography, and then you interpret the results. Then you try to mutate the protein using site directed mutagenesis. Then you try to express the mutated protein, purify the mutated protein, then you do an X ray crystallography, and then you compare these two structures. A very good lab will take at least 5 years to do this. A lab with all the money and all the infrastructure and all the brains that are required. The same thing today, you as a biologist, with a love for coding, can do that in 5 to 10 minutes, maybe 1 hour. That's the max. So that's how AI and ML is changing the way we look at data, and that is why omics technologies are the next big thing in science. But coming back, so this was the background. In the next 10-15 minutes, I will tell you how to use these techniques to understand a disorder, because it's good to have a technique, right? It's just good to have laws that don't walk on the don't always wear helmets while using a bike, but then you have to keep fines and policemen to ensure that people are wearing their helmets. So I work on a disease that is known as hepatic encephalopathy. Now, this is a strange disease because it starts from the liver, but it expresses itself. So, the manifestation of the disease is in the brain. So, it's, to put it very simply, if the liver fails, the brain gets impacted. And you see, in all our life, something that we really appreciate is memory, right? When I started off my talk, I told that, okay, I have very pleasant memories of Mumbai and my stay at IIT Bombay, and I think it is my second home. All the reasons is because I have a memory of Bombay, right? The moment you wipe that memory, it doesn't make sense whether this college is situated in Bombay or Boston. I won't have any specific like or dislike. So memory is, as Oscar Wilde said, is the diary that we all carry about with us, and it impacts the way we behave, the way we think. So if somebody gave you a very bad memory, you would try not to go back to that place or to that person. And if something has a good memory, you might go back to that person or place time and again. And memory is primarily controlled by the hippocampus. So when I'm talking of hepatic encephalopathy, so this is a loss of brain function that occurs when the liver is unable to remove toxins from the blood. Now we know that we produce a lot of metabolites and toxins. One of the very important toxins that we produce is ammonia, and this ammonia is converted to urea in the liver. And when this toxin is not removed from the brain, it from the blood flow, it reaches the brain. And this disease is basically called hepatic encephalopathy, and it may occur very suddenly or it may develop slowly over time. So again, uh, we need biomarkers for this, because right now the only way of recognizing this is through psychomotor tests. And if you think that HE is not common, then take a look at this data. So this data was taken for minimal hepatic encephalopathy, and which is the most covert form of the disease, which means you are suffering from the disease and you don't even know that you have the disease, right? Because many a times we have issues with our digestive system, and we don't give it much importance whether we have the same issue in our brain, and there is always a stigma associated with brain. So if somebody is having a broken hand, they will happily go to the hospital to fix it. The moment you tell that, I think, you tell your friend that, okay, I think you are having some psychological issues, he will take it as an insult, he or she. मैं पागल हूँ, how dare you, right? This is how we react to any psychological or neurological disorders. How could you say that I'm mad or I'm crazy? And that's one of the reasons why diseases such as minimal hepatic encephalopathy, they don't go undetected. And if you see in this center, what they did, they tried to see how many people with liver diseases have MHE. So this is not percentage of the entire population. It's the percentage of the population who reported liver diseases and came to the gastroenterology department of the hospital. And then the doctors thought, let's have a psychological test to determine whether this person is also suffering from hepatic encephalopathy. And you see the numbers are staggering. In a place like Pune, it's 93% of all patients who arrived at the hospital. Mumbai, 42%, Delhi, 58, Kolkata, 62, Chennai, 31, Bengaluru, 67. So, and this is a very recent study, 2019, just prior to COVID. So, you cannot negate the fact that people suffer from minimal hepatic encephalopathy. And worldwide, 80% of patients with liver failure develop minimalized HE in India, and out of them, 30 to 50% develop overt HE. Overt is a very strong form of the disease. Here you start having really, you forget, you just behave like an Alzheimer's patient. You cannot, you have, I mean, you have a combined impact of Alzheimer's and Parkinson's, wherein you cannot walk properly, you cannot grip things properly, you forget things, you have complete loss of memory at times, and sometimes people go into coma where they fail to recognize anyone and they go into this deep coma stage. So this is a very staggering problem that needs to be looked into. And how do we have it is mostly cirrhosis of liver, and that comes also from alcohol abuse. And if you see, go back to these, you see these cities like Bengaluru and Pune, lots of IT sector people, they work hard and they party harder and they go on binge drinking nights, and that destroys their liver. They, most of them have sedentary lifestyle, and which again uh, causes fatty liver, and all these together leads to rise in the ammonia levels in the blood. So you have cirrhosis, alcohol abuse, drug intoxication, people who take opioids and benzodiazepines as medication, and sometimes as a drug abuse, right? Apart from that, bacterial and viral infections, they can also impact. And the major source of ammonia is our gut. So the food we digest, the food we eat and then we digest, that is then broken down and it releases a lot of ammonia. But under normal circumstances, the liver takes care of it. It turns into urea, and then would be excreted via the kidneys in the form of urine. But if it does not, it goes to the brain. And ammonia being a very small molecule, right? It's NH3. Blood brain barrier can do very little to stop it. So when the NH3 goes inside, it starts causing diseases. And how it happens, if you go to the basic biology or pathophysiology, the molecular level at it. So NH3 goes inside and it triggers the glutamate to glutamine cycle, right? And there is a enzyme called glutamine synthetase, and What it does is to trap the NH3. Okay, glutamate will be produced to glutamine, and glutamine is then taken up by this presynaptic neuron. And in the presynaptic neuron, there is another enzyme called glutaminase. Now, glutaminase will now convert glutamine to glutamate, and then glutamate will be released in this junction of the two synapses. Once it is done, in the postsynaptic neurons, there are excitatory receptors, primarily NMDR. There are other receptors, but NMDR is the primary receptor that picks up glutamate, and this is a excitatory receptor. Now, what happens if I ask you that, okay, all of you, today you have 30 minutes of time to play? You will be very happy. Now I increase it to 1 hour. Again, you will be very happy. 1 hour, 30 minutes. Perhaps some of you will be happy. Others will tell, "Arey sir, kafi tired lag raha hai." Right? And then if I keep on increasing it, okay, the next 24 hours you have to play cricket or football. I'm pretty sure most of you will pass out by the time it's 10 hours or 12 hours. A very athletically active person, perhaps till 18 or 16 hours. And that's what happens to the neurons. When they are continuously excited, they suffer from energy deficit. There is high oxidative stress, and that leads to apoptosis and necrosis of the cells, and this leads to brain dysfunction. So now you would say that, okay, we know everything. Why would you? So why do we do proteomics? We know the disease. This goes wrong. So that's what brings us to the lacunae of the study. So if I block these receptors like NMDA receptors, it's a very good, right? I am asking you to play 24/7. The best way to do is put me in Arthur Road jail. Okay, that guy is in jail, so he will not ask people to play 24/7. But problem is NMDA receptors are very, they are very important, and they have lots of other work, and normal brain function is not possible without NMDRs. So you cannot block NMDRs. So another way is, okay, everything that's happening is happening due to high levels of ammonia. Now, why do we not reduce ammonia in the gut? Why don't we eat food that minimizes the production of ammonia? This is a very good pharmacological intervention, but it is not very effective, and it is often met with severe side effects. It's like they will ask you not to eat most of the food that you eat today. You have to completely stop your protein intake. Mostly you have to survive on carbohydrates, and that also specific type of carbohydrates. And this leads to deficiency, malnutrition, and again, so you don't die due to a brain disease, but die due to malnutrition. Again, not something that we as a scientist would prefer. So to find this, so we need to relook into this entire HE, hepatic encephalopathy pathogenesis, and to find new proteins that might be impacted, right? So whatever we know is fine, but we now need to have more information so that we can we can stop this disease from getting aggravated. And the answer, could proteomics give us the answer? Now, this is to tell you what goes on in a typical brain. So what you see here uh, is the control brain, and these are neuronal tracings. So we have traced the hippocampal neurons, and you see they are pretty big, branched out like a good tree. So this is the root of the tree, this is the branches of the tree, and they are pretty big. What happens in case of minimal HE or moderate HE is you find that these trees are shortened. If you again look at these cells, Now I will tell you, in these cells are very globular and healthy. Now here you can see many cells are shrinking. These cells are shrinking because these cells are now treated. So HE stained sections showing increased neuronal cell death in hippocampus of HE rats. So what we wanted to do is, so we so this is primarily to show you that it's not just the behavior that gets impacted. There is a anatomical reason behind it. The cells are really inside the neurons, inside the brain regions. These neurons are really dying. So the death of these cells are leading to the disease manifestation. Now, idea was, can we study the proteome of the hippocampus, uh, and then try to identify if there are different proteins? So this is the hippocampus of the rat brain, and we wanted to see that if I just take out the hippocampus, extract all the proteins that the hippocampus is expressing, both during normal and disease state, can we actually have a better understanding of the disease? So this is what we did. We took two groups of rats. One was the control rat, another was the group that had moderate HE. We took out the hippocampal tissue, we extracted the proteins, we do a QC check of the proteins, then we do a trypsin digestion of the protein, then we clean up the peptides. So I'm not getting into the techniques much. So we do the, after trypsin digestion, we clean the peptides, put it through an LC-MS instrument. We take out the raw MS data using database and statistical searches, network analyses, using AIML to understand the DEPs. We try to understand what went wrong in the diseased group. So what we found was, we could identify 2,175 proteins in the hippocampus, out of which 1,895 proteins was statistically validated. This is something that perhaps is out of purview of this lecture, but idea is, although you identify all the proteins, we have to apply some statistical tools to make sure that the proteins that we are identifying are not false identifiers, or we are not falsely implicating some other protein. So we apply some statistical tools to validate these protein numbers. Then, out of these 1,895 proteins, 47 proteins showed significant dysregulation. When I'm talking of dysregulation, I mean 41 were upregulated and 6 were downregulated. So there were 41 proteins which showed increased expression in the diseased hippocampus. 6, the expression had come down. And when you talk of this is the volcano plot that we usually use to, so these are all the proteins, the more center line you find. So these are the proteins that were, they showed variations, but those are biological variations which we don't take into consideration. We are talking of these proteins, FAC1, HBB1, fetchivin A. So these red ones are the upregulated, the downregulated ones are marked in violet, and these are the proteins that were downregulated, these were upregulated. Now, based on literature search, we further found out that, okay, of all these proteins, do we have any information of these proteins? Did other groups study? So, and also another thing that we did was, we did a network analysis, enrichment analysis, to see these dysregulated proteins, are they impacting any other, what pathways are they interacting? So that's what we see. So you see, gliogenesis is a pathway that is getting highly impacted. Also, you can see modulation of the chemical synaptic transmission, regulation of myelination, astrocyte differentiation. So all these neuron specific processes are getting impacted during MOHE. And then we took up the top 12 differentially expressed proteins, and we found that these 3 proteins, focal adhesion kinase 1, CD9 antigen, and tetraspanin 2, these proteins are again very strongly correlated with the physiological manifestations of the disease. And we have some, so this is what research is. You find data, then try to make sense of the data using both AI/ML tools and also using literature, that okay, are these proteins specific to the brain, and what are the changes that other people have seen. Now, a very interesting one was, if you just, okay, so the next slide is validation. As I told you, that we have to do targeted proteomic studies to again find out the, to validate the actual proteins. What we did was, we studied the FAC1 expression in dentate gyrus, CA1 and CA3 regions of the hippocampus, and we found in, okay, not only did the protein express, the protein expressed differentially in the different regions of the brain. Similar studies we did for CD9, a protein, and we using, fluorescence, immunofluorescence. We wanted to study what are the different changes that we see in the brain regions. CA3 dentate gyrus. So these are part of the DG is dentate gyrus. CA1 is cornoamynous 3 and cornoamynous 1. These are parts of the hippocampus. So using these tools, we wanted to study what are the specific expression patterns. So we are now validating what we found in proteomic studies. We are trying to validate it using a targeted study. Next, we did the same study for tetraspanin 2, and again we found increased expression in MOH. Now we again go deeper. Now our idea is, okay, since these proteins are expressing in higher amount, can we find a pharmacological inhibitor of this protein and see whether injecting this drug or injecting the molecule will impact the disease pathogenesis. So what we did was, we took up focal adhesion kinase 1. It is a cytosolic non-receptor tyrosine kinase, and it has very strong role in cell adhesion, cell migration, and survival. And if you would remember, we were talking about neuronal growth. So FAC1 is highly enriched in neuronal growth cones. What we did, we took a specific drug called Y15 and we injected it into the cells, into the rats as a post treatment. So can it reverse the impact of MOHE? And this is a very hot, this is a very recent study that we did. We are still in the process of validating it and publishing it. So when we found that if we can treat the rats with this Y15 drug, these are pyramidal neurons, and you see these pyramidal neurons are showing a lot of recovery after the treatment. So this treatment procedure can actually help you reverse the damage that was done by MOHE. So when you do a co-treatment or a post-treatment with Y15 analog, a Y15 drug, since we are blocking this FAC1 protein expression, you can reverse the impact of MOHE. So this is how, we can actually take proteomics data, try to do AI/ML based search strategies to find out our candidate proteins for therapeutic interventions or biomarkers. The biomarkers part I did not keep because most of my studies on COVID-19 using proteomics was on biomarkers. So maybe some other day we can talk about biomarker discovery. So using biomarkers or using these proteome expression changes as tools or methods to study how a disease progresses, right? We can effectively use it to treat a disorder. And just must thank you. So this work was done in collaboration with multiple labs. One was lab of Dr. Papia Acharji from Department of Zoology, BHU, Professor Sanjeeva Srivastava from IIT Bombay, as well as Professor S. K. Trigun at Banaras Indian University. So I would like to thank all the members who contribute to the study and also to the funding agencies who provided us. I think with this I will stop. This is my QR code for my LinkedIn. So anybody who's interested can connect with me on LinkedIn. We can have a discussion of whatever your career doubts are, whether you want to learn more about proteomics. Um, I'm ready to help you out. And also, one more invitation is to join this program. So anyone who is interested in learning more about proteomics and how you can use proteomics and AI in biology and medicine. So we are organizing this GYAN program. So GYAN is a Ministry of Education approved uh, scheme. And here we are having Professor Graham Ball, who is a very renowned expert of AI and ML and its use in biology. As one of the faculties, you will be the primary faculty. You're also having faculty Professor Sanjeev Srivastav from Department of Biosciences and Bioengineering, IIT Bombay, and also me myself from Department of Zoology, University of Allahabad. So this will be from November 25 to 29th, University of Allahabad. You can register yourself. Of course, the seats are limited, but if I feel that you are really interested and you can go through these 5 days of really strenuous training, we will shortlist you and we will send you an invite to join this program. So with this, I will conclude. I think I can take questions right now.
Yes, thank you, sir, for such an informative and an interactive session. With this, we come to an end with session 2 of the webinar. I thank Arup, sir, for giving us the basics of central dogma and the difference between the concept of proteomics and genomics. The concept of proteomics was well explained using examples as well. Thank you, sir, for giving us the exhaustive information on the different types of proteomics and how these different types are connected to genomics as well as the different methods of analysis. Sir, you also give us an interesting overview of the areas being studied in proteomics as well as the challenges faced in the data analysis. The role of AI as well as the machine learning was also explained in an easy and a well-lucrative manner. Sir, we'll begin with the question answer session. Participants can post their doubts in the chat box. Sir, there is one question which is currently in the chat box. I'll read out to you. What is the statistical validation of proteins and which tools can be used for the same?
Okay, so you see, I don't want to confuse you on proteomics on day one, because the more deep you go, the more confusing a technique becomes. Now, let me tell you like this. So, every sample, when we study, there can be, uh, we do replicates, right? We do replicates in the sense each sample is run on the instrument for twice or thrice times, 3 times. So, it can be duplicate or triplicate. Now, suppose I run a sample 3 times and I find all these 3 different times, because it is an instrument, it will give me a variation in results. So sometimes it will give me 2000 proteins, sometimes it will give me 2100 proteins, sometimes it will give me 2000, maybe 2010 proteins, right? And all these proteins, the numbers are not only varying, then the variation will also come in terms of the proteins they are identifying, right? So sometimes it may be ABC, next it can identify ABD, next it will identify ABCDE. So what we do is we take this entire data set and then we apply tools such as, first of all, we normalize the data, right? Yeah, and these are completely statistical terms. We normalize the data. We also do something known as, which is missing value imputation, where we try to find it, since it's a big data set and any data that is missing will lead to statistical analysis problems. We try to do missing value imputation. That okay, in a particular experiment, in 10 replicates we found protein A to be present. When we repeat the experiment, we find protein A is present in 7 samples, not in 3 other samples. Now there can be 2 reasons why it was not found in 3 other samples. One, the sample was actually not present, the protein was actually not present on those 3 samples, or the protein was not detected by the instrument. So statistical tests, delve into these different kinds of strategies, where we try to make the data set simpler. We try to make the data set less confusing. So that's why whenever you see the number of proteins discovered and finally the number of proteins that we take forward for our statistical analysis is always less. So we cut down on the dubious proteins that, okay, this protein is sometimes expressing, sometimes not expressing. So we need to, we apply stringent laws. So it's just like in your college, there is a 75% rule or 80% attendance rule, because your college feels that, okay, those people who have not attended 80% or 75% of the classes, they do not even know the subject well enough to take the exam. So that's, I mean, in brief and in very simple terms, I try to tell you. Of course, these things are not very simple. It will take time and a separate lecture, perhaps 4 to 5 hours of lectures, to tell you in deep what are the statistical tools. There are many instruments. There is MaxQuant. There is one software called MaxQuant. There is MS Fragger. There is MetaboAnalyst. So these are very specialized softwares that we use. So this is not just random softwares. We also use a lot of coding using R and Python. So those coding languages are also important. We have libraries in R and Python, such as Bioconductor, and there are packages by which you can analyze data.
Thank you, sir. Gokul Shelke, you had raised your hand. Could you please put forth your doubt or question which you have? Okay. Sir, the next question is the experiment on genetic neuronal disorder on mice and humans. Will it show different result?
So, you want to study mice versus humans in a particular disorder?
Yes, sir.
Uh, of course, it will show difference. You see, if you compare mice versus mice, that's a fair comparison. If you compare mice versus humans, you will find lots of differences. Um, but yes, you might find some commonalities. But point is, most of the human diseases that we study in mice do not suffer from Parkinson's or epilepsy or Alzheimer's. We create disease models. And how do we create disease models? We create disease models just like the rats in my experiment. They were healthy rats. How did I damage their liver? I injected a chemical called thiacetamide in their peritoneum, and that chemical damaged the liver, right? So, but a normal human might get liver damage due to hepatitis, uh, drug abuse, alcohol abuse. So, of course, so whenever you do a study on a plan an experiment, you will study either a disease model, and there you have to do mice versus mice, or rat versus rat, or Drosophila versus Drosophila, and for humans you have to find human tissues. Significantly harder to find human tissues. That's why most of the studies that we see uh, are on mice and rats. I answer the question? But overall,
Yes, yes.
overarching, if you tell what other will it be extremely different? No, it won't be extremely different. The devil will be in the details. And when you're trying to design a drug, uh, details are what that makes or breaks a drug. So you can try out on mice, you can find few pathways, then you have
Mhm.
to actually validate. So whatever I have shown you was on rat. Now, can I take Y15 as a drug in humans? No. It will take at least 5 to 10 years to validate the same. I'm going to find similar results in humans, and then maybe 5 to 10 years more to use it as a drug on, do a randomized control trial study on humans. So, but yes, the baseline, the very rough draft of the experiment needs to be done on animals and cell cultures. That's the utility of wet lab that we do.
How can the integration of computer-aided drug design and proteomics enhance the drug discovery process, particularly in identifying and validating potential therapeutic targets and biomarkers?
Okay, computer-aided drug design will not help you in biomarkers, but of course, it has huge implications when it comes to therapeutic targets. As I told you, with the advent of tools such as AlphaFold by DeepMind. Just go and Google AlphaFold DeepMind. You would find how deep and profound their impact has been. I'm just telling you. So suppose you want to create a drug. You want to find a drug against a misfolded protein. Computer aided drug design will only work if you have a proper structure of that protein. Now, this proper structure of the protein earlier used to be done using tools such as X-ray crystallography and NMR and stuff. But now you can just take a protein sequence, mutate whatever sections of the protein you want to mutate, give that sequence to AlphaFold. AlphaFold will generate a 99% with 99% accuracy a protein, that misfolded protein. And then, of course, you can use tools such as the computer-aided drug design, molecular docking softwares. Then you can, check your entire drug library against that misfolded protein and see how it is interacting. So, of course, drug discovery has changed a lot. What for therapy for biomarkers, what you need to do is use AI ML like random forest classifiers, support vector machines, and other ML tools, machine learning tools to go through that data, right? You humanly, it's not possible to check for variations in maybe 5 crore protein expression patterns, but through AI/ML, they will learn the patterns very easily. And once they understand the pattern, that okay, in epilepsy, this is the pattern. In normal, this is the pattern. Immediately, it will tell you, okay, for this protein looks like a biomarker. One more important study thing about biomarkers is biomarkers can be of two types. One is prognostic, another is predictive, another is diagnostic. So if you are talking of a predictive biomarker, it tells you whether you might have the disease. And they will tell you maybe 5 or 10 years before the disease onset. That is a different class of biomarkers. Then there is a diagnostic biomarker. Diagnostic biomarker will tell you whether you have the disease or not, right? Most of the biomarkers people find today are diagnostic. Then there is another set of biomarkers known as prognostic biomarkers, and prognostic biomarkers tell us whether after having the disease, what will be the course of your disease. Like the work we did for COVID-19, we wanted to find severity biomarkers after COVID, whether will you get a severe COVID or you will be let off with something asymptomatic or mild. So again, it's a very, very tricky field, and lots of money to be made, to be honest, if you can do good science and find a proper biomarker for any of these diseases. Like as I was discussing, Matthias Uhlen's group in Sweden, they find biomarkers for cardiovascular diseases. Now think of the startup potential. You can find biomarkers for cardiovascular diseases, and you can sell those biomarkers not only to to normal people, but you can ask medical insurance companies that okay, if a particular client is having this biomarker, they are more prone to heart disease, you can charge more premium for them. Or if a particular client is having less of this biomarker, they are not likely to get cardiac diseases, so you can charge less premium from them. So the opportunities are huge in when it comes to omics field. What is holding us back is primarily lots of regulatory issues, lots of issues regarding finding good samples. Proteomics as a technique itself is very nascent compared to genomics. Genomics is much more stable as a technique. Proteomics is a much more nascent technique. We still do not do whole length sequencing of proteins. We would do shotgun sequencing using mass spec. So newer technologies are coming in, and maybe 5-10 years from now, the way softwares are developing, the way technologies are developing, we will be in a much better condition to work on the true biomarkers, that is predictive biomarkers. To tell someone that okay, you might have Alzheimer's, you might develop cardiovascular diseases, you're prone to ischemic stroke. So this is, the best way to prevent a disease, to stop a disease, is to prevent the disease altogether.
Can the process of post-translational modification in proteins affect proteomes study?
Yeah, we do. There is an entirely long branch of proteomics where we study post-translational modifications. There is glycoproteomics, there is phosphoproteomics. Yeah, and we do that. I mean, we do that with full energy. So one thing is, in discovery proteomics, it can tell you with prediction, it can predict whether this particular protein was phosphorylated or not. And then in phosphoproteomics, you can actually see which amino acid residues was phosphorylated. And yeah, so the answer to this question will be yes, it does.
So there are no questions from our side. Thank you so much, sir.
Thank you, Pushka, and thanks to all of you. Around
Yes,
140 people have joined.
so approximately 190 people.
Okay, I can see 140, maybe 50 left.
Yes,
But
yes,
okay, thank you for listening, and do connect to me on LinkedIn, and we can discuss more.
sure, sir. So let us
Thank you.
begin with the validity session. I, Mr. Pushkar Bhojne, on behalf of Patkar-Varde College and the entire faculty of Department of Microbiology and Biotechnology, deem it a great honor to propose the vote of thanks to all those who made this webinar a resounding one. I begin my vote of thanks by addressing our appreciation to the management of Chikitsak Samuha, Chief Education Officer Dr. Mala Kharkar. I will extend a hearty vote of thanks to both the speakers for taking out their precious time for enlightening participants and making this webinar a great success. I take this opportunity to thank Dr. Jitendra Kumar Sinha sir for his efforts towards unlocking the code, deciphering the molecular secrets of brain disorders. And will also like to acknowledge my gratitude to Dr. Arup Acharji sir for his enormous efforts towards unlocking the mysteries of the disease brain, the power of proteomics and big data analytics. I also thank Mrs. Uchita Dalvi, Chief Coordinator of Sciences and Coordinator of Microbiology, Biotechnology Department, Mrs. Anupama Tomar, Co-Coordinator Department of Microbiology, Mrs. Neha Colasso, Co-Coordinator Department of Biotechnology for their constant support. Behind every successful webinar is a team of dedicated individuals who work diligently behind the scenes. My gratitude goes out to the teachers who ensured the seamless execution of this event, allowing us to come together in a virtual space where ideas and knowledge flow freely. We're also grateful to a sponsoring organization, RUSA. This event wouldn't have run smoothly without a technical team. Last but not the least, thanking the participants who have turned up in such a great number from all over the country and for the active participation throughout the webinar. The participants will receive their participation certificate through the mail once they have filled the feedback form, the link for which has been posted in the chat box. Please note, the link will only be active for 30 minutes. In closing, I want to thank each one of you once again for your unwavering dedication. Thank you.